So let's welcome our next speaker, Abubakr Shafique, who is currently a postdoc in Toronto Metropolitan. He will introduce the work related to fairness of foundation models.
Thank you so much. You can see my screen. Okay.
Yes.
Thank you so much. Thanks for the introduction. I'm a postdoc at Image Analysis in Medicine Lab at Toronto Metropolitan University, working under the supervisor Dr. April Khadimi. So today I'm going to present our work, How Fair Are Foundation Models, exploring the role of covariate bias in histopathology, which was previously accepted at FAIMI workshop. So I'm honored to have this opportunity to share our findings and engage in discussion with the esteemed researcher here. Our work mainly explore the fairness and covariate bias in the foundation models for computational pathology, highlighting the importance and considerations for equitable AI deployment in healthcare. So these are the outlines for my presentation. I'm going to go over with the introduction and then talk about the domain shift, and we're going to define the fairness in the healthcare and what foundation models we used in our study and how did we prepare our data and what methodology we used to compute the covariate shift and some results with the conclusion. So first of all, we know that it all starts with the biopsy and then the data preparation, even though we know there are standard protocols for the staining the slides, but still due to different sites and different centers and different conditions, there are still some variations in the staining pattern, which are differing from one center to another center. So traditionally, the pathologists, they do analyze the glass slides under the microscope and render some diagnosis. But now we have in with the advancement of model tools, we can digitize the whole slides and we can perform some automated tasks. With the digitization, we know there are its own unique challenges. As we can see, when we do digitize those glass slides, we end up with gigapixel high resolution image with cellular level details. As we can see, with the hardware with the hardware we have, it's not that is not able to compute the whole slide as a whole. So what we do is we divide the big image into a smaller tiles in order to process them. Then we have a domain shift problem since we are scanning these whole slide. So usually what we do is we are in research, what we are doing is we are taking a data from one center and then we are setting a data like 80% for the training and then 20% for the testing, but both data are essentially from the same center. And then we are happy that, oh, our models are rendering 97% results or above 90% result. But when the same model is being tested on the data from another center, the model is then producing sub 60% results. This is one of the major bottleneck in the deployment of AI models across the clinical workflow because usually when we go out of domain distribution that the model have not seen, the models perform poorly. So there are mainly four types of domain shifts, mainly the covariate shift, which is due to the difference in data acquisition and the patient demographics. And then there is prior shift, when the source and the target domains are pulled from different data sets. And then there is a posterior shift, which is changes in clinical definition or labeling criteria, such as when the new grading standards are being made or there is some evolving disease presentation or the emergence of a new subtype. And then there is a class conditional shift when the data characteristics for a specific class differ from source and the target domains. So in this study, what we have done is we took the same glass slides, but then we have scanned it with the two different scanners. So in this case, we have kept the staining protocol the same. So the only differentiating factor here is the scanner. So as we can see, the same glass slide being scanned with the two different scanners, but their color histograms are very different for the same patches from the same location. So even for the pathologists, if they observe the same slides under the microscope, I mean they can see the same cellular morphology. But for the computers, even the slide color differentiation can make a huge difference. So as even with the naked eye, we cannot see the difference, but uh, for the computer, the models, they can see the huge difference. So here are some of the examples for the patches that are extracted from the same spatial location for the glass slides that were scanned from the two different scanners. Let's define the fairness in the healthcare. Usually, fairness in healthcare in terms of AI analysis refers to the principle of the particular algorithm and the model that to provide equitable performance across different patient groups without any bias such as race, sex, age, ethnicity, or socioeconomic status. But here in this study, we also interpret the fairness as the robustness of the AI model to the covariate shift introduced by the different scanners, which can also be the factor in unequal performance in the healthcare settings, which can also help us understand if the models actually are learning the cellular morphological features or they are just learning the scanner signals or the distribution based on the cohort or the signatures of the scanner. So for the fairness and the generalization, recently we have seen in the emergence of the foundation models, which have been trained on very large scale of the data because of this the self-supervised learning, the advancement in self-supervised learning. We no longer need the to label the large amount of data in order to train the foundation models, which itself is a cumbersome task and which may introduce the annotator bias or variability. However, since the self-supervised learning do not need the label data, we can use the large GPU clusters and large deep architectures, and we can use all the data that we have in order to train the foundation models. And with these foundation models, they have the capability of strong generalization, and they can be used with little or no fine-tuning for the various downstream tasks. So in this study, we have used various foundation models with different architectures, mainly the vision transformers, but the size of the models varies from 7 million parameters all up to the 1 billion parameter, the biggest model available in the literature. And all these models are trained on different types of different data set with various different organs. So we have like a variety of the model that we can compare with. So in our data set, we have data available from two different centers. One is OICR, which is Ontario Institute of Cancer Research, and the other is University Health Network in Toronto. So both of the slides were scanned with their in-house scanner, which was Aperio 82. And we have 111 slides, and we extracted 4,952 patches, which were at 20x magnification, and the patch size was 512 by 512. Similarly, with the UHN, we have the data was scanned with their in-house scanner, Aperio ScanScope, and then we rescanned it with uh, our in-house Vision Tech Sakura scanner. And the patches were extracted at the same spatial location after the image registration. So in our dataset preparation, as we can see, we scanned the slides, and then obviously we know that they are not at the same spatial location after after we have scanned. So we had to do the image registration since they are the same slides. It was uh, pretty accurate when we do register those slides using the fast sample consensus method to have the corresponding matching points. And then we only used affine transformation, so we don't bend the tissue or D-shape any of the tissue. So we just used the rigid transformation. So once we have the rigid transformation, we can use the spatial location. So we extracted the patches from the same spatial location, as you can see here, and we use the foundation models to extract the features. And then we do compare the feature based on the mean square error, KL divergence, and we also compare the cluster using the CH index. So these are some of the results for the MSc as we compare the patch pairs together, like one patch from the Sakura scanner and the same patch from the Apereo. So we compare patch by patch, and we have a distribution of the MSE. As we can see, some of the model like HIPT has the uh, lowest feature difference, and even the foundation models, they have some kind of feature difference as computed by the mean square error. So one of the reason to compare with the mean square error is it is not dependent on the length of the vector, so we can compare different models together as each of the model is generating different length embedding vector. So in uh, other metric like KL divergence, we can see the distribution comparison of the feature distribution, how they are compared with each other. So there is still some kind of a difference for each of the data set, as we can see, and also for the clustering. We can see even like the foundation models like Virchow also has some kind of higher clustering index. So usually the CH index, we prefer the lower CH index, but in our case, we usually we prefer higher CH index so that the clusters are well separated. But in our case, since we are dealing with the same cellular morphology, we want them to be closer together. But as you can see, we have a higher CH index. That means that the clusters are far apart. And here are some of the differences between the data set, because as we know, we have two data set. We also compare across the data set how much difference the model compares from one data set to another. So there are some significant differences as well in the MSC, but there are like less difference in the model HIPT, which also has lower MSC and KL divergence, and the CH index as well. So also we can visualize the embeddings in the 2D space based on the TSNE plots. As we can see, all these are the corresponding patches from two different scanners, but we can still see a clear differentiation between the scanners for each model. Here we can see that the features are jumbled up. That means the model is considering that as a one cluster, but in most of the models we can see there is a clear difference. Here we have mixed signals again here for the gigapath, the uni we have clear mixed signals, but for some other cases we have a clear distinction. So this is for the OICR dataset, and this is for the UHN. Again, we can see a clear distinction between the scanners. Again, the HIPT is like jumbling up all these corresponding patches. The uni also is performing really well based on two different scanners, and there is a little bit a distinction between two scanners here. So in conclusion, we have seen that even the foundation models are prone to the domain shift, and HIPT, despite being the smallest model among all, has shown the lowest MSE and KL divergence. Among the big foundation models, Uni, Gigapath, and Virchow 2 are exhibiting relatively lower representation shift. Uni is smaller than Virchow 2 and Gigapath, but still it exhibits a similar performance. So that means even the model architecture is bigger. We need to keep that in mind, that it does not really change the performance, and the strong data augmentations are recommended in order to train the foundation models, and we do really need extensive evaluation strategies to include in the domain variations when we are training the foundation models. Thank you very much. I would like to thank my co-authors and my PI, Dr. April, for this study. And if there are any questions, you are welcome.
Perfect. Thank you, Abubakr. I guess at the moment, no questions just yet, but there we go. As soon as I say that, that always happens. There's one question which we'll take. It says, do you have any insights into why some models are more robust than others?
I think it was all in training strategy. Some use like a good data augmentation strategies, and they use very diverse data from different scanners. So I think this is one of the reason that some models were doing good and the others were not.
Thank you. I think there's some other more questions about not understanding dementia, but maybe you can just reply to them in the chat as well. But thank you so much.
Sure, thank you so much.
I'll hand it over back to Emma.